My name is Nurul Ain binti Muhammad Liki. My number matric 23 DBT 20F1012. I'm from DBT 3A. Today, I will do lab simulation for the experiment bacteria identification. In this experiment, I have to do 4 lab simulation, which is gram staining, catalysis test, coagulase test, and lastly oxidase test. For now, I will do lab simulation for gram stain. This is the procedure for do lab simulation gram stain. First of all, I will explain about the gram stain. The gram stain is the most frequently used stain in a clinical microbiology laboratory and is usually the first step in identifying bacteria. Based on difference in cell wall component, bacteria are categorized as either gram-positive, which is standard purple, or gram-negative, which is stained pink. In addition to the gram reaction, the morphology and configuration of the bacteria can be observed. Gram-positive occurs in chest or gram-negative root. First step we will do, which is click on the Bunsen burner and pass the slide gently 2 or 3 times to the flame. Do not overheat because can cause destruction of the cell. And second, flood the slide with crystal violet for 1 minute. And then rinse with H2O. After that, flood the slide with iodine for 1 minute. And then rinse with H2O again. Next, decolorize with alcohol for 5 to 10 seconds. And then rinse with H2O. After that, flood the slide with safranin for 1 minute. And then rinse with H2O again. And lastly, view slide under microscope. This is the result for lab simulation gram stain. Second, I will do lab simulation for catalysis test. This is the procedure for do lab simulation catalysis test. First of all, I will explain about catalysis test. The catalysis test is used to differentiate Staphylococcus, which is catalysis positive, from Streptococcus, which is catalysis negative. The enzyme catalase is produced by bacteria that respire using oxygen and protect them from the toxic byproducts of oxygen metabolism. Catalase positive bacteria include strict aerobes as well as facultative anaerobes. Although they all have the ability to respire using oxygen as a terminal electron acceptor. Catalase negative bacteria may be anaerobes or they may be facultative anaerobes that only ferments and do not respire using oxygen as a terminal electron acceptor. The first step, which is drop H2O2 on slide. Second, pick isolate colony. And then mix bacteria into H2O2 . And we can see from that it will have bubble for the Staphylococcus aureus catalyst, positive or negative. This is the positive. The positive have the bubble. And then dispose of the toothpick. Dispose of the slide. Then we do the same for this one. Mix bacteria into H2O2. And for negative, we can see not have bubble. And third, I will do lab simulation for coagulase test. This is the procedure for do lab simulation for coagulase test. First of all, I will explain about coagulase test. Coagulase is an enzyme produced by cells that convert soluble fibrinogen in plasma to insoluble fibrins. Cells actually produce two forms of coagulase, solute and bound. This benefits the bacteria in the host in that bacteria coated with what fibrins might be protected from the host immune system. First of all, drop water on the slide. And then pick isolated colony. And then mix bacteria into water. Next, dispose on toothpick. And then add 1 to 2 drops of rabbit's plasma. After that, rub slide and wash for clumping. And lastly, dispose of the slide. And then repeat the procedure for step errors. Drop the water and then pick bacteria mix to H2O2. And then dispose of the speak. After that, add 1 to 2 drops of rabbit plasma. And root slide. Wash for clumping. This is the positive. This positive have the bubble. And lastly, dispose of the slide. We are done for do lab simulation coagulase test. For the lastly, I will do lab simulation for oxidase test. This is the procedure for do lab simulation for oxidase test. First of all, I will explain about oxidase test. The oxidase test to aid in identification of bacteria that produce cytoserum C oxidase and enzymes of the bacteria electron transport chain. All bacteria that are oxidase positive are aerobic and can use oxygen as a terminal electron acceptor in respiration. This does not mean that they are strict aerobic bacteria that are oxidase negative, might be anaerobic, aerobic or facultative. The oxidase negative result just mean that these organisms do not have the cytoserum C oxidase that oxidizes the test reagent. They may respire using other oxidase in electron transport. Bacteria generally characterized as oxidase positive include Neisseria and Pseudomonas. Genera of the Enterobacteriaceae family are characterized as oxidase negative. First of all, I have removed dry slide from packet. After that, pick an isolated colony with a sterile toothpick. And then group bacteria onto slide. And then wash the color. If the color change a blue, so that means the bacteria is positive. And then dispose of the speck. After that, return the procedure to another bacteria. Root bacteria onto slide. And then the color is yellow, so that's mean negative. And lastly, return. So this is the negative 2. Dispose the slide, and we are already done.